You're discussing the distribution of these cholinergic receptors in the brain. I'm not too concerned about that. Part of the problem here is that this information tends to change over rather short time spans. But you should be aware, as you look at these illustrations, of the ubiquity of both the nicotinic and the muscarinic receptors. They're all over the place. They're in virtually every region of the brain and contributing to the various functions that those regions are involved with. We'll talk more about that in our discussion later. All right, let's talk a little bit about the amines. Remember the amines, the first thing that should occur to you is that the amines utilize the slow pathway. We want to classify them chemically a little more precisely. We usually differentiate the catecholamines, that have a catechol group, from other types of amines. The catecholamines all derive from tyrosine. And the pathways for synthesis of dopamine, norepinephrine, and epinephrine are very, very similar. I don't know if you've been keeping up with Oliver Sacks. Oliver Sacks was a renowned neuroscientist. He has terminal brain cancer, or terminal cancer of some sort now, and he's sort of chronicling his thoughts, and I think he's written 2 memoirs. He too spoke at Nova a few years ago on the main campus. But there was a movie associated with one of his first books, Robert De Niro, Awakening, something like that. You might remember that movie. The individuals, now that was a slightly different disease, but the individuals were in a type of psychosis that could be inhibitory. But in any case, and we'll talk more about dopamine in a moment, the miracle drug, unfortunately short-lived, it doesn't act very long, for a very long period of time, was something called L-DOPA. And L-DOPA is a precursor not only for dopamine, but for these adrenergics as well. So dopamine, norepinephrine, and epinephrine are the catecholinergic amines. And then there are a few others there are many others, but a couple others we should be familiar with. Certainly histamine will come up in our discussion. We've already talked about histamine and being involved in inflammation. It functions as a neurotransmitter, a hormone, an immunological cytokine. It has a number of functions. And the other one we should know that is a non-cholinergic amine is serotonin. As I say, this is not the acceptable term anymore. The term we should all be using is 5-hydroxytryptamine or just 5-HT. The serotonin is derived from its earliest observed physiological effect, and that was to regulate peripheral blood flow. Cellular blood, tonicity, regulation of the smooth muscle and the peripheral vessels. But of course, it does many more things, and we'll talk about that in just a moment. But for now, just be aware that 5-HT, or serotonin, is an adrenergic neurotransmitter. And of course, the little cartoon here is intended to suggest that all of these are at least thought to play a role in the one's emotions, affect. But some of the evidence for that may be a little specious, as I will suggest a little later. Now, these 2 brackets are important because I do want you to notice a couple of things. Now, we haven't talked about neuroanatomy yet, but this is the brainstem here. We'll talk about the 3 parts of the brainstem. And what you'll notice in the brainstem is that there are focal centers or focal points of radiation for both noradrenaline and serotonin. So if you look at the illustration on the left, there are adrenergic. What's the word I'm looking for? Adrenergic origins in the caudal-raphe nuclei of the brainstem. A couple things we need to talk about here. But my point is that these neurotransmitter systems originate in the brainstem. That's going to be true for both the adrenergic and serotonergic systems. And I do want you to be familiar with some of these terms too. So let's talk about caudal-raphe. Caudal just means tail. Raphe is midline. Tail, the sort of midline tail. The term nucleus, when we talk about the nervous system, refers to a cluster of neurons in the CNS. So the term nucleus is just a cluster of neurons in the CNS. We'll have another term to describe a comparable cluster in the peripheral nervous system. So the caudal nuclei, and this one you should know as well, the locus coeruleus. It's a very important adrenergic focal center in the brainstem, site of radiation. And from these points, especially from the locus coeruleus, you can see that adrenergic pathways radiate throughout the brain into the cerebellum, into the cerebral cortex, both anterior and posterior. The focal point for the serotonin brainstem focus for the serotonergic pathways is also in the caudal nuclei, but there's one up here called the rostral. In general, we just say that the serotonergic pathways are associated with the raphe nuclei. Again, raphe meaning midline. And they too, those serotonergic pathways you can see extend into the cerebellum, into the basal nuclei, into the thalamus, and then again into the entire cerebral cortex. So I just want you to anatomically, whenever we talk about these neurotransmitter systems, just keep in mind that they are very critical, important foci for these adrenergic systems in the brainstem. So we've already talked about the mechanism of action. You are familiar with this graphic. We looked at it before. All of these adrenergics utilize the slow, or for our purposes, G protein signaling pathway, a second messenger signaling pathway. Put a note up here, don't confuse the alpha and beta adrenergic receptors with subunits of the G protein. So in other words, this has nothing to do with an adrenergic alpha receptor. If this is epinephrine or norepinephrine, this whole thing could be a G protein receptor, but it's also an alpha adrenergic receptor. That has nothing to do with this, which is a subunit of the G protein. Don't know if that makes things clear enough. I just want to make sure you don't confuse the two. Talk a little bit about dopaminergic systems. Once again, you can see there are important radiating sites for dopamine in the brainstem, but unfortunately, I guess it's a little more complicated than the others. And I'm going to do my best to summarize the pathways that you need to be responsible for. But before we get into that, make sure you understand that dopamine is another aminergic neurotransmitter utilizing the slow synaptic pathway, G protein pathway typically. Dopaminergic pathways are critical for, we think, certainly motor function. We're going to talk about dopaminergic pathways and their importance in regulating motor function. But they also probably play a role in modulating emotion, mood, affect as well. Perhaps the most hype, the most PR associated with dopaminergic activation is the reward pathway. Basically, dopaminergic pathways are involved in every aspect of motivation, risk-taking, your drive to do whatever it is you're driven to do. And I don't want to know about it. So with that in mind, let's talk about 2 or 3 fundamental pathways. I think for now we'll just talk about 2 of these important pathways, and they're sort of highlighted here. We're going to, there we are. I've named them, or at least we will name them in some fashion like this. The first is this critical motor pathway. We'll call it the nigrostriatal pathway. Nigro, because it originates down here in the substantia nigra of the midbrain. Substantia nigra just means dark matter. Remember the precursor to melanin? Sounds like a good quiz question. What is the precursor to melanin? Tyrosine. And so we get the coloration from the sun sort of stuff. So it's called the nigrostriatal pathway because it goes from the substantia nigra down here to the basal nuclei up here, and a portion of the basal nuclei is called the striatum. You'll have to learn. We'll talk about the basal nuclei a little later. But for now, I just want you to know that the nigrostriatal pathway, and there it is, it's this pathway on top here, is the pathway that's most involved, for example, in blood regulation that goes awry in Parkinson's disease. So what's the essential problem in Parkinson's disease? Well, in terms of the manifestation, but neurobiologically, what's going on?
The lack of control.
Well, that's true too. The lack of dopamine, so it's not the lack of dopaminergic cells, but it's the lack of dopamine specifically in the substantia nigra. And one thing you're going to find out as you approximate that point in life when you are getting close to colonoscopies is that you're going to be reaching for something in the dark one day and it's just not in focus. And you get closer and it even gets less in focus, and that's called accommodation. It's called presbyopia. You lose the ability to focus as you age. That's a function of age. There's not a darn thing you can do about it. Just as you get older, you're going to lose accommodation. Likewise, if we had the means to do it, we could measure the dopamine levels in your substantia nigra, and they too decline with age. It's just that in Parkinson's, the decline is abnormally accelerated for some reason. But if you get into your late 80s or 90s, most everyone is going to have. It's the rare individual that does not have some type of tremor associated with dopamine. This particular pathway, this nigrostriatal pathway, the problems in the basal nuclei. All right, we'll talk more about that later. Just want you to be familiar with the pathway now. This other pathway is the one that gets all the hype. That's how do I have it labeled up there? The dopaminergic mesolimbic reward pathway. This is the fundamental reward pathway. Even addictions such as opiate addiction trigger this pathway. Food addictions, internet addictions, whatever your addiction is, that if you would, please list them before you leave. Whatever it is, it's going to be involving this particular pathway. So this is the one that gets a lot of attention for that reason. And what you'll notice is it's more anterior. It goes to a region called the nucleus accumbens, and then up to this part of the brain, the frontal lobes. What do you do with your frontal lobes? I'm asking you because I have no idea. Mine haven't been used in decades. But what do you normally use your frontal lobes for? I think so. Executive function. You also do your planning, your anticipation here. So I think you can maybe see the connection. If you get this between reward, gratification, motivation, anticipation, planning, all of that sort of fits together. Here's the substantia nigra, so I didn't have to spell it out. Yeah, there are a few other systems. We won't worry about those. Those are the two primary systems. We'll come back to those as we talk about the regions of the brain later. Now, in this pair of illustrations we have, as we can see, this is not very clear. This needs to be redone. I'll work on it. But this is the presynaptic neuron. This is the presynaptic neuron. The purple here is the synaptic cleft. And here's what, make sure you label it because students sometimes have trouble with these illustrations. The postsynaptic cell is down here. So don't be confused by this perhaps not perfectly rendered illustration. So the purple is the synaptic cleft, and then this is the postsynaptic membrane. And these pink purple things are the receptors in the postsynaptic cell membrane. And then over to the left, we have the cholinergic synapse. This is a cholinergic synapse. This is an aminergic synapse. Is that clear? Cholinergic, aminergic. So let's look at the one on the left first. Here's our presynaptic cell that's going to be releasing acetylcholine in a voltage-gated, calcium-dependent fashion. Right? You know what I mean by all that. And so the acetylcholine is released, and it diffuses all over the place, and it's sufficiently high in concentration. It's going to bind to the postsynaptic receptors. And if it does, then we're going to have, in this case, what type of response will we have? What type of receptor is that? Well, but more specifically, it's a nicotinic. So you're going to have the fast, you're going to have immediate depolarization. This is what you're going to find. How do I know that? Because here's my receptor and here's my channel. Muscarinic receptor wouldn't look like that. All right. And here's the deal. Here's what I want you to get from this illustration is that in the cholinergic synapse, the acetylcholinesterase, which is the catabolic enzyme that breaks down the acetylcholine, it lives, it resides, it makes a living in the synapse. It's just, again, like a little Pac-Man floating around, gobbling up acetylcholine. So whether or not acetylcholine will go from the presynaptic or the postsynaptic cell depends upon the relative abundance of acetylcholine and acetylcholinesterase. If there's much more acetylcholine, we're more likely to get postsynaptic membrane depolarization. But if there's more acetylcholinesterase than acetylcholine, then neurotransmission probably will not occur. Is that clear? So a couple of things to keep in mind here. Make sure you know what the function of acetylcholinesterase is and know where it lives. It acts through synaptic enzymatic degradation of the neurotransmitter. And it's simply the relative abundance of the neurotransmitter versus the enzyme that determines whether or not neurotransmission will occur. Yes?
So the cholinesterase is constantly being made all the time?
Yes, and it's a fairly high concentration in the synapse, and the concentration in most synapses does not change very much. And so it's just all the time in there, ready to gobble up whatever it can. And then, of course, we talked about this before. When it breaks down the acetylcholine, it breaks it down into choline and acetate. So both can be taken back into the cell and recycled, but they're no longer functional neurotransmitters at that point. What would be the effect of an acetylcholinesterase inhibitor? Would an acetylcholinesterase inhibitor enhance or depress neurotransmission? All right, so everybody make sure you understand that an ACHE inhibitor will enhance neurotransmission. I asked you a question earlier about what are the primary medications for Alzheimer's disease, what are the FDA approved? Most of them are acetylcholinesterase inhibitors. Do they improve the symptoms? Do they have a great effect on Alzheimer's disease, Alzheimer's symptoms? No, you're talking about 2 or 3% improvement at most, so it's not a very effective therapy. But that's the mechanism. Any questions about that? Because now we want to focus on the aminergic synapse. And it works a little differently. So over here, very different. We have a presynaptic cell. Here's our synapse again. Here's a postsynaptic cell. Here's the postsynaptic. And by the way, why aren't these ionophores? Why aren't these ion channels? Why isn't this a receptor channel? Because all amines use the second messenger signaling, so they don't work the same way as the nicotinic receptor, so we don't have that morphology. All right, so the presynaptic cell is going to release, let's say it's norepinephrine as is shown here. It binds to the postsynaptic cell membrane receptor, eliciting some type of postsynaptic response. And then here's the deal. In general, let's just say that there is no primary mechanism for removing the amines from the synaptic cleft. Rather, and this is what's a little bit hard to sometimes get a handle on, the catabolic enzyme that breaks down noradrenaline, dopamine, and so on, is located in the cell that released the neurotransmitter. It's located in the presynaptic cell. And you can see what MAO is monoamine oxidase. It oxidizes the neurotransmitter. All right, so how can such a system work? And it's dependent upon this thing right here. Oops, let me make that a little more precise. What is that? That is a membrane transporter. That is called a reuptake transporter. And what is its job? Pump the neurotransmitter in the cleft back into the presynaptic cell where it's broken down and recycled and retooled and so on. So, a couple questions for you here. What would happen if we inhibited the reuptake transporter? That would increase the concentration of neurotransmitter in the synapse. What is an SSRI? What do those terms stand for? Selective serotonin reuptake inhibitor, operating under the premise that if you inhibit the activity of this thing, you're going to have one of those green dots. They're not going to be hitting back into the cells. Their concentration is going to increase inside the cell. You're increasing neurotransmission in that fashion. All right? That's one. Yeah.
How does the norepinephrine get released from the protein?
In most cases, you're just talking about, that's again calcium-dependent exocytosis. So there's no. In other words, we have the action potential.
I misspoke. In number 2,
right?
The norepinephrine is released from that protein.
Yeah, that's a little bit misleading. Once again, whether or not the neurotransmitter will bind is simply a function of the neurotransmitter to receptor concentration. In other words, the relative concentration of these. If we have the more neurotransmitter I have in the synapse, the more likely I have that there will be binding to the receptor. So if I increase the number relative abundance of neurotransmitter in the synapse with a finite number of receptors, I'm increasing the probability that there will be thousands of these, but increasing the probability that any one of those is going to bind the neurotransmitter. This can happen, and the catch and release that you see here happens, but it's not important. It's simply the analogy I used to use was like fiddler crabs in a marsh, and one of your classmates in a previous class used to make fun of me to such an extent that I stopped using that analogy. But that's not the primary. This doesn't affect the concentration very much. It's this, and that thing up there we have yet to talk about that affects neurotransmission.
Is there competitive inhibition on those proteins?
There can be. There could be any number of substances which would block this. For example, we talked about what do we call the receptors for epinephrine and norepinephrine? Well, they're adrenergic, you're exactly right. Alpha or beta, so an alpha or beta blocker would be such a creature.
Is the reuptake transporter
Is it what?
Is it always just constantly running?
Yep, it has to. If it doesn't, you have problems. You have some of the symptoms that were described earlier with respect to another inhibitor might arise. And we'll talk about that a little more because I'm going to talk about how antidepressants work and so on. So the other thing, before we leave this, so we can tie it all in a bundle here, MAO is the catabolic enzyme that resides not in the synaptic cleft, but up there in the presynaptic cell. And its job is to break down the neurotransmitter. Suppose it stopped doing its job. Suppose it stopped breaking down the neurotransmitter. Think about your basic stoichiometry and chemistry. And the green thing is building up inside the cell. What's going to happen to the activity of the reuptake transporter at that point? Would it increase or decrease, and why? Decrease. Why would the transport rate decrease? The concentration gradient is becoming more difficult. As the concentration increases inside the cell, this thing's having to work harder to pump pump the neurotransmitter back in. So I guess my question for you is, what would an MAO inhibitor do? So an MAO inhibitor would increase or decrease the concentration of neurotransmitter in the cleft. Increase, right.
So is this a passive, the reuptake transporter, is that passive? Or is it VEP?
In the clinical literature, you sometimes see a reference to passive reuptake. In reality, that describes pharmacologic events more so than what's actually happening in this protein. So I would not encourage you to think about passive reuptake in the membrane. This is an active transporter. And that's a little more complicated. It depends. The ability of this transporter to pick up the norepinephrine or the amines in general depends upon a variety of factors. Only in part, ATP is involved to some degree, but there are other factors involved as well. But I think the key things that I want you to keep in mind is, again, what are the effects of a reuptake inhibitor and an MAO inhibitor on the concentration of neurotransmitter in the adrenergic synapse. Now there's another, in that table we mentioned another catabolic enzyme called COMT. It doesn't live there. It doesn't live where the MAO does. It lives over here somewhere. It typically lives way off of the synapse. Can anybody think of a reason why that might be useful to have catabolic membrane enzymes away from the synapse? There's a high concentration of adrenaline in your blood right now. Why is it there? Do you have any sort of gland that releases adrenaline in your blood? Yeah. And you would think the adrenal gland would be responsible for most of the adrenaline in your blood, but where do you think most of it actually originates? From the brain. Now, what these COMTs do, again, we living way off the ranch, living somewhere else, is they help to disperse, to reduce the peripheral concentration of these psychoactive, these drugs, that these neurotransmitters that may otherwise affect neurotransmission. So these just basically are scavengers. They basically reduce the total level, the tonic level of neurotransmitter, circulating level of amines in the brain, the CNS. But they don't seem to play a role in local regulation of neurotransmitter.
So do this COMT only live in the brain, or do you have
As far as I know, they're only the CNS. All right, so we've talked about the amines. Let's talk a little bit about the amino acids. You already know an important. You already know the mechanism of the amino acids. What's the mechanism? How do are they fast or slow? Fast. So all the amino acids are fast. We have several classes here to refer to. The glutamate, aspartate, glycine, GABA. These are all examples of amino acid neurotransmitters. If they are glutamatergic, remember we looked at that figure earlier. We looked at GABA and glutamate synapses on the postsynaptic cell. And we said that glutamate is the most abundant excitatory neurotransmitter, and GABA is the most abundant of the inhibitory neurotransmitters. And again, that's a function of the postsynaptic membrane receptor. And we'll talk about these in more detail in just a moment. Unfortunately, these get a little more complicated than the others that we talked about. The two primary classes of glutamatergic receptor are the NMDA and the AMPA. The NMDA, N-methyl aspartate, is probably the more important of the two, and it's a little complicated in its mechanism of action. We'll look at a little animation in just a moment to explain these. But this is important. You should be aware of the NMDA receptor because it mediates most of the glutamatergic excitation in the brain. Remember, glutamate is the most prevalent exciter. Another important thing to keep in mind here is that boy, this thing works entirely different. We could devote an entire year talking to how this thing operates. But let's just simplify it and we'll look at the animation. The NMDA glutamatergic receptor is both voltage gated and ligand gated. Both have to happen before it will open. And it's even more complicated than that. You should also keep in mind that the NMDA channel is important in learning, long-term, something called long-term potentiation, a learning process, and therefore it's critical to keep in mind a lot of issues involving cognition or impaired cognition involve problems with these channels or at least with glutamatergic neurotransmission. And it often works in association with the AMPA receptor. By the way, NMDA is an agonist. So let's make sure we understand that. Very complex receptor. The NMDA receptor is a glutamate receptor. It binds glutamate. It's called the NMDA receptor because that's just one very specific agonist that binds it. And therefore, it's unique in that regard. But it's a glutamate receptor, so glutamate binds it, then what happens? It's a fast, that means there's an ion channel. What type of channel is it? What type of ion passes through it? You can see it's a cation channel. It's a cation channel, fairly non-specific. But here's the deal. It's chiefly a sodium channel, so it's going to be excitatory. But the learning component, the way that the receptor contributes to the ability of your cells to learn and remember, is more calcium dependent. So, NMDA channel, glutamatergic receptor binds glutamate, causing a fairly non-specific cation channel to open. Chiefly sodium, being excitatory, but in terms of its physiological function, you will often see it called a calcium channel because calcium mediates the learning response. Yeah.
Sodium and calcium can be outside.
Yep.
So is this channel both ways?
Yep. And that's often the case with these cation channels. So when that happens, when we talk about a cation channel being excitatory, why is it excitatory and not inhibitory if both potassium and sodium are passing through it? Anybody have a theory? You folks are usually pretty good at theories.
Sodium passes through more often.
As well, what?
But you said that it's mainly, it passes all cations, you said it's mainly a sodium channel, right?
Yes,
Sodium passes in more often than potassium.
Okay, that's right. I think, yeah, that's a good way of saying it. The sodium ion concentration gradient is more significant, greater. It's a greater gradient than the potassium gradient. Therefore, you get the predominant depolarization. But remember, this is the only significant intracellular cation. You've got several others. Yeah.
So this is a non-specific channel. So is this NMDA channel that's opening non-specific for those cations? So you're saying it's not specific to which one it refers to, but it could be any of those three different
No, that's what I was trying to stress. So this is definitely a glutamatergic channel. So it binds glutamate. And then on binding glutamate, it allows cations through according to their relative concentration gradients.
But it'll allow all three of them through at once?
Yep.
There's not three different NMDA channels.
No. And that principle is true. Remember we talked about the ion channels earlier, and we said that we sort of specify their ability to allow only one ion through. In this case, a variety of cations are passing according to the relative concentration gradients. So we can say that this is in effect an excitatory channel because of the sodium current, but I just want to let you know that in your literature later, when you talk about issues involving glutamatergic excitotoxicity and neurotransmission and problems with cognition, they'll describe it as a calcium channel because it's the calcium and the calcium pathways that are involved in what is called acquisition and retention, your ability to learn something and then remember it for a long period of time. Maybe this little animation will make it clearer. If not, I did my best.
AMPA and kainate receptors are relatively simple. They require only glutamate binding to open the ion channel, which leads to membrane depolarization. In fact,
So remember what she said, AMPA. The AMPA channel is open. All right, so the AMPA is just a single cation channel there. All right, so let's keep going.
excitatory neurotransmission. Activating NMDA receptors is more complex. Both glutamate and glycine, each of which bind to different sites, are required. Moreover, in resting NMDA receptors, the ion channel is blocked by magnesium. Membrane depolarization by activation of a nearby AMPA kainate receptor displaces the magnesium,
Depolarizes that.
allowing the NMDA ion channel to open. Calcium influx through NMDA receptors contributes to long-term potentiation, a phenomenon that may be involved in long-term learning, synaptogenesis, and other neuronal functions.
That help, yeah.
In that video, you can see potassium coming out.
No, you wouldn't, but it is this. So it's just a non-specific, and there was something else missing there too. For example, she called it a calcium channel. It's not a calcium channel. The calcium channel is important only for the cognitive aspects, the learning. And if we have time, we'll talk about the LTP later. We really don't have time right now. But anybody have any questions then about the glutamatergic receptors and how they operate? Yeah.
So the NMDA channel doesn't become activated until
Bind to different sites are required.
Sorry. There we go.
glutamate binds the AMPA channel, which gets the magnesium off the NMDA channel.
Yeah, this induces remember, for the NMDA channel to open, it has to bind the ligand, the glutamate, and there must also be depolarization of the membrane. The AMPA channel, most commonly, not always, but most commonly, is responsible for the membrane depolarization, which is necessary for the NMDA channel to then do its thing. But you'll notice, we could argue that it's not really a glutamatergic channel. Why? Because what else has to bind to it? Glycine. Yeah. So let's not overcomplicate this. Let's call this a glutamatergic channel that acts as a non-specific cation channel if the membrane voltage is depolarized.
Is the glycine binding on the AMPA channel or the NMDA?
NMDA, if I didn't say that, I meant to say it, on the NMDA.
What's binding the AMPA then?
I'm sorry?
What's binding the AMPA then?
The AMPA, this is a little bit complicated too. The AMPA will generally accept either glycine or glutamate. And if it's usually glutamate because the glutamate is responsible for initiating the depolarization that the NMDA channel uses. Glycine is inhibitory. Glycine acts like GABA. And this is just a little slide to have you remember that NMDA is important in learning. This is sometimes called the learning channel. You thought it was on cable, but it's actually in your brain. And we can make mice smarter by increasing the concentration of NMDA receptors. You don't. What do you use to increase your powers of concentration? You use coffee? I thought the common answer was Adderall. I have a little stand set up in the hallway if you want. Now, I was just, this dates I think from the 1960s, but this is an advertisement for benzodiazepine that acts upon this next class of receptor, the GABA receptor. But what is the implication? I was trying to write on that. Hold on just a second. What's the implication here? This was medical advice provided to doctors. Is no one offended by this? What's the implication of the picture? She'd happily do her housework if only what? She had some drugs. And the benzodiazepines act upon the GABA receptors. Now, so here's the thing. We'll think about that, but here's here's the GABA receptor here. So GABA is a amino acid neurotransmitter that binds to this chloride channel. When it binds to the chloride channel, chloride's at higher concentration outside the cell. It's bringing a negative charge in. Is that hyperpolarizing or depolarizing? Hyperpolarizing. So this is an inhibitory channel. So if we can upregulate that, which is what the benzodiazepines do, you can set her free. I just think that's highly obnoxious and absolutely. But again, times are changing. So
What do you mean upregulate?
I'm sorry?
What are you upregulating the
No, you are upregulating or increasing GABAergic neurotransmission. GABAergic neurotransmission is excitatory or inhibitory.
Inhibitory.
So you are alleviating the anxiety associated, at least that I have, with housework. We have a couple of minutes here. We'll come back and talk about serotonin because it's a fairly complex topic. So the proper term is 5-HT or 5-hydroxytryptamine. This is a non-catecholaminergic amine. Most of it, surprisingly, is in your GI tract, not in your CNS. And so we'll talk a little bit about what it does there. It's present in platelets. It seems to play a role in mediating, for example, coagulation. And you probably know with our discussion of SSRIs that it's thought to play an important role in mediating mood, affect, emotion, and so on. And we've already looked at those sites of radiation in the brainstem, the Raphe nuclei in the brainstem. There are a number of isoforms of the 5-HT neurotransmitter, the receptor. They are all second messenger except for the 5-HT3, which is an ionophore, relatively uncommon. And it's certainly important. There are all kinds of studies implicating serotonin, for example, in obsessive compulsive disorder, aggressive behaviors, all kinds of things. But of course, it gets most of its attention with respect to depression. This is an antidepressant. And there are a number of classes of antidepressant. I just want you to be aware of what that acronym or abbreviation suggests. There are also similar reuptake inhibitors for norepinephrine and serotonin, SNRIs, for example. They're the older class of tricyclics and tetracyclics, just refers to their chemical conformation that also act as reuptake inhibitors. The SSRIs are preferred because these have more side effects. These have more toxic responses, and that's true for the MAOIs too. So normally you would prescribe the SSRI, and only would these classes be prescribed if the other class did not work. The MAOIs have a number of adverse effects, but at least you understand the mechanism of action. Before we move on, I just want to point out a study that came out this month, and then maybe you can discuss it when we come back at the next class. Our serotonin levels are a chief determinant of depression, and they're all kinds. This is a 2015 study. That is in mice. If you have mice that have no ability to produce serotonin, they weren't depressed. You might ask yourself, how do you know if a mouse is depressed? They behaved more composedly. They behaved normally under stress. They responded to other antidepressant drugs like normal mice. You might ask yourself, how do you know if a mouse is normal? And in studies in humans indicate that about 2/3 of those taking SSRIs do not improve. Others have shown no effect. Most studies show the effect is comparable to a placebo. So we'll stop there. I'll let you reflect upon that and maybe talk about it next time.